So what we're going to talk about today, this particular unit, and really going forward, is stuff that's really applicable to everyday life. It's stuff that you do on a regular basis. So even though we are 6 weeks into the class, that first 6 weeks is really learning about the microorganisms. It's understanding how they grow, it's understanding how they work, and the last 9 weeks are all application, are all understanding how this is impacting you. So we're going to take today and Thursday as well, and we're going to talk about how we control the growth of microorganisms. Today's lecture is really going to be physical and chemical methods of control on an inanimate surface. And then Thursday, we're going to discuss what do you do when those microbes are in your body. So how do we control the growth when they're in the body? Things like antibiotics, okay? So we're really talking about, now that we understand how microorganisms grow, now that we understand how they divide, now that we understand what they're causing, a little bit about that, how do we then take all of that information and say, how do I want to kill them at home? You know, I know how temperature affects enzymes, so how do I use temperature at home to control the growth of microorganisms? Okay? People have been doing this kind of stuff for hundreds of years. Think about food preservation, okay? You get a can of peas at the store. What is that can full of? Besides the peas and the water. What other compound? Salt, okay? What do we remember about salt? What does salt do to living cells? Drains out all the water, right? It's going to suck out all the water. So when you buy canned products, there's typically a large amount of salt that's in there, or a fair amount, or some other chemical that's meant to draw out the liquids, because it's used as a preservative. It's used as a preservation technique. You buy frozen meals at the store. They're full of salt, okay? Lots and lots of things that you can buy down those main aisles at the store are all full of things that are just preservative in nature, okay? You ever looked at a hot dog? It's full of all kinds of nitrates and nitrites to make sure it lasts longer. Lunch meat, same exact thing. It's all about food preservation, and it's all about controlling the growth of microorganisms. That's it, okay? When it comes to controlling disease, all right, we do this all the time as well. You have a flu. Now, you got a stomach bug. Stay at home, okay? Why? Why would you want to stay home if you're sick? Number one, you want to get better. You're contagious. You don't want to be spreading it to everybody else. Every time you're coughing, you're sneezing, yes, you're spreading it, but guess what? It's all over your hands. So everything that you're touching is potentially spreading that organism from one person to the next. You know, that's a lot easier said than done, okay? Me. Do you know what it takes to find a sub to cover me to not come to class? It's like an act of God. It's ridiculous, okay, to try and find a sub to cover 3 hours of microbiology. It's not easy. And then I'm here and I'm teaching to all of you about spreading disease and all this kind of stuff. So it really stinks when I'm sick and I have to stand up here and go, I know I'm talking to you about all these bacteria and spreading of disease, but I couldn't stay at home today because they couldn't find me a sub, and well, we couldn't cancel the class because that's like out of the question. So no, you're all going to sit here and watch me, and I'm sick, and yes, we're going to try and make sure nobody else gets sick as well, okay? But we do this all the time, okay? You see somebody and they're coughing and hacking up a lung, and you're like, just stay away from me. Just go over there, or you get out the hand sanitizer, all right? It's all about controlling microbial growth. Drinking water. Drinking water is one of the biggest ways, if you drink contaminated water, that you're going to end up getting sick, okay? So your drinking water is all been filtered. It's all been extensively tested to make sure it doesn't contain microorganisms. The minute there is some sort of failure in that system, at any point along the way, they're issuing a boil water alert, okay? And there's a lot of fail-safes to make sure that that doesn't happen, all right? Because you don't want to get sick. But think about all of the things you do on a daily basis to control the amount of microorganisms in your life. You know? You take that milk carton out of the fridge, you put it right back in the fridge. You don't, oh, more milk, disgusting, okay? Live bacteria in there, all it does is just breed, okay? You make sure things are cold. You got leftovers, you put them in the fridge, right? So you do things all the time to make sure that we're controlling microbes and not getting sick, okay? With this particular lecture, a lot of this is stuff you've heard of. This is nothing new. But it's really about connecting our terms and connecting our microbiology to everything that you've learned already. All right? So I do want to say, bless you, that the terminology here is really important. When you see side on the end, that means to kill. But you also have to look at the root word that's in front of it. Okay? So bactericide. Bacteria is in front of it. It's talking about bacteria. That's a chemical that's going to destroy bacteria. Keep in mind it only destroys living cells. It has no effect on any sort of dormant state. So if there are endospores present, a bactericide is not going to do anything to it. A fungicide can kill fungal spores, hyphae, yeast. Viricide, chemical that inactivates viruses. Notice it says inactivates here and not kill. Remember, our viruses are not living, okay? So that viricide has the ability to inactivate the virus, to denature that capsid. You can have a sporicide. Sporicides are actually capable of destroying bacterial endospores. When you see these terms, germicide and microbicide, these are the two of the most generic terms that you're going to get in terms of microbial control. It basically means we kill some bacteria, but it doesn't mean they kill everything. So if you see something and it says this is a germicide, it could kill 2 species of bacteria. It could kill 100 different species. There's no guarantee with that term as to what it's doing. It's very, very generic in nature. It simply means we kill some bacteria. It could be killing a lot. It could be killing a little. You don't really know, right? So in terms of this list, think about our diversity chapter. Okay, we just talked about that on Thursday. Basic bacterial cells are the easiest things to kill. They die very easily and very quickly. So something that's a bactericide doesn't have to be really stringent in nature, okay? But fungal yeast, fungal spores, hyphae, sporicide, spores, viruses, those are harder to destroy. So we're going farther on the list in terms of these guys are typically more harsh chemicals versus something that's a bactericide is typically, can be fairly light in nature, it just depends. If something is sporicidal, bacterial endospores are the hardest things that we have to kill, besides prions, but prions are a whole other category, okay? If something is sporicidal, you can believe it that that's a pretty harsh chemical, okay? And guess what? If it gets rid of bacterial endospores, it's strong enough, it's going to inactivate viruses. It's going to get rid of fungi, yeast, hyphae. It's also going to get rid of bacteria as well, okay? So the sporicide here is the one that's really going to be the toughest one, and it's going to kill any sort of bacterial endospores that are there. If you see germicide or microbicide, it could mean it's a fungicide. It could mean it's a viricide. You don't really know what it's getting rid of. Okay, it's a generic term. On the other hand, when you see the ends stasis or static, that basically means to stand still. It does not mean we're killing it. It means it's no longer growing and dividing. So a chemical that's bacteriostatic or a method that's bacteriostatic simply prevents the growth of bacteria. Preventing the growth. Fungicide inhibits fungal growth. Microbistatic, something that's used to control microorganisms but is not meant to kill them. So static keeps them where they're not growing and dividing. Think about our terms on temperature. I'll keep coming back to that one a lot because it's easier to understand. When we talked about temperature, we said lower temperatures slow down enzymes. Higher temperatures speed up the enzymes to the point where the enzymes denature and it gets rid of it, okay? So higher temperatures are cytal to bacteria because they destroy enzymes. Lower temperatures, on the other hand, send it into a static state because we're not killing the organism. We're not doing that at all. We are simply making sure that the enzymes aren't functioning, it's not growing. Think about this. You take your leftovers and you throw them in the refrigerator. Whatever bacteria are on that food when it goes into the refrigerator are still on there. They're not dead. You're just cooling down the temperature to send it into a static state. If you left the leftovers on the counter, that warm enough temperature is going to still allow the enzymes to function, which allows metabolism to work, which allows the organisms to grow and divide. Does everybody see the difference between a static versus a killing function? Static means they're not going anywhere. If what you need is to kill them and instead you send them into a static state, that's not really doing you any good. Okay? Putting your raw chicken breast in the refrigerator buys you a few more days, but it doesn't mean you kill whatever bacteria was on there. When you take that chicken breast out, you still have to cook it. Okay? So when we say microbial death, what do we mean by that? Yes, we talked about the bacterial growth curve. We talked about that death phase, okay, where bacteria are dividing in a logarithmic manner, albeit a slower manner than which they grow. Death is not instantaneous, all right? They don't all die all of a sudden. But the key here is that if a cell can no longer reproduce under ideal environmental conditions, it's in a state of death. It doesn't mean it's instantly dead. It may die 10 minutes from now. It may die an hour from now. But if it cannot reproduce, and it's got all the nutrients, and it's got the right temperature, and it's got the right pH, if it can't reproduce under those correct circumstances, it's in a state of death. Okay? Death when you're adding a chemical, death starts when that threshold of that microbial agent is met, and then it continues in a logarithmic manner. So think about this. You want to clean your toilets, okay? That's a nasty job. Nobody likes cleaning toilets, okay? What do? You get out whatever toilet bowl cleaner of your choice, or you can simply dump bleach down the toilet, okay? You can use one of those nasty brush things, okay? It's up to you. You add it in there. You swirl it around. If you flush the toilet right away, your effectiveness in cleaning it is actually minimal in terms of getting rid of bacteria, because you only let it sit on there for a certain amount of time, okay? However, you squirt that stuff on there, you move it all around, okay, fine. Just let it hang out. Let the next person who comes in to use the restroom flush the toilet. Because the longer that chemical sits in the bowl, the longer you're going to make sure we're down here, we're killing everything that's in there. Okay? You got something nasty you want to clean. You dump bleach on it. Okay? You're trying to clean, I don't know, toys, cups, or who knows, whatever you want to clean at home. You're going to add bleach to it. Okay? You don't stick it in the bleach and immediately rinse it off. You've done it no good. You have to reach a certain amount of the chemical for a certain amount of time, and then we start to kill things off. Cells don't all instantly die. It's going to take time for all of the cells to have access to the chemical and for there to be enough damage that it actually causes death. Okay? You marinated chicken. Okay? And you want to reuse that marinade. That's possible. Okay? But what do you got to do? You should boil that meat. You should cook your chicken separately, boil that marinade, and you need to let it boil for a good 5 minutes to know that you've killed everything in there. It's not bring it up to a boil and shut it off. You're under a boil water alert. They don't tell you bring your water up to a boil and then shut it off. You have to let time do its thing. You have to make sure that whatever that agent is that you're applying, whether it's a physical method or a chemical method, has enough time to kill all of those cells that are in there. We making sense with that? Okay. how do we do microbial control? There are tons of different methods of microbial control. Physical methods are things you apply to an item, okay? So whether that's heat, whether that's UV light, okay, or ionizing or non-ionizing radiation, whether that's sending it through a filter, okay, or some sort of mechanical removal method, physically pulling it out, okay? That's a physical method of control. Your bread is moldy a little bit, but you really need that piece of bread. You tear off the little bit of mold and you're like, all right, I'm going to eat the rest of it, okay? In theory, that's just a mechanical removal method. Yeah?
Like that, so it's like, you can't eat that. Just put it in the toaster oven and you scrape it off.
It's fine. It's all okay.
You don't die.
It's, yeah, exactly.
Like that.
You haven't died yet, so it's probably okay. But, yeah, it's just a matter of whether you want to do it or not. Yeah, no.
No.
To each their own. Have I done it? Yes, I have. Okay, the kid is like, I have to have this. I'm like, oh, that does. I see a spot on there. I don't want. That's what I want. Fine, just cut that sucker piece off and hand it to him, and you're not gonna die from it. It's fine. Yeah.
My grandma's about 75, and she's like, well, if I didn't die when I was young, I'm not gonna die now. So what's all this got to do with anything?
See,
It's funny.
you know, what's the worst that's gonna happen? In most cases, you're gonna get some food poisoning, and you're not gonna feel very good for a few days, and then you'll be fine. You'll tell everybody I got a stomach bug, and it's all okay. All right. But you can physically treat something, or, depending on what it is, you can add a chemical, okay? Understand that you're not bleaching the food that you're eating, okay? So, keep in mind what you're adding and when you're adding them, okay? Typically, chemical methods are going to be reserved for inanimate objects that we're trying to treat. Physical methods are more going to be for things that are going in your body, foods, that sort of thing, okay? Understand your terms. Notice that killing bacteria is not really using them. We're trying to destroy them, okay? So when you see that an item is sterile, that means there are no possible living organisms anywhere on it. We've removed the endospores, we've gotten rid of the viruses. There is nothing living anywhere that's on this thing, okay, if the item is sterile. So we can do this in a number of different ways. Most of these are physical methods of control. There are chemicals, on the other hand, that are so harsh in nature that they are capable of sterilizing. So we have all different methods. Again, it's going to come back to what you're trying to treat. On the other hand, if you disinfect something, this simply means you're eliminating most of the pathogens. Okay? Means we're getting rid of most of the bacteria. Disinfection is kind of a misnomer. People think I disinfected something, so that means there's nothing on it. That is absolutely not the case. Disinfection simply means you're bringing down the level of the pathogens that are present. So that could mean you seriously cut it down, or that could mean you do a little bit of work. Okay? Two terms affiliated with disinfection, so you understand disinfectants, like Lysol, okay? An inanimate object or surface. If you have an antiseptic, antiseptics are chemicals that are safe for use on living surfaces. So disinfectants are harsh enough, they're inanimate objects only. Antiseptics are not as harsh, and they're safe for use on living things. Now, you can have a chemical that is both. Okay? Alcohol, for example, can be both an antiseptic and a disinfectant. You go to get your flu shot or something like that, and they rub that little alcohol prep pad on your skin. It's an antiseptic when it's used on a living surface. You want to clean benches in a lab? Alcohol is actually a really good disinfectant. Okay? Most research labs use ethanol basically across the bench. Okay? We did for a little while in lab until we realized, we have open flame and sometimes people aren't paying attention and it's straight up alcohol and that's just not a good mix. Okay? But it works really well. Okay? So it just depends on what you're trying to treat. Antiseptics go on living surfaces, disinfectants go on inanimate objects. There can be some crossover there. When you hear that something has been pasteurized, typically what's happening in pasteurization is that we're reducing the items that are specifically involved in food spoilage. And so typically with pasteurization, it's something that you're actually going to eat or ingest. And it's briefly heating something up. So pasteurization is actually the use of heat, specifically for microbial control. Guys, there are tons of random terms that you can see in everyday life, okay? So if something is sterile, there's no organisms on it. I don't care what the other term is. If it's not sterile, that means there's some level of microbes that are present. So whether you see the words decontamination, degerming, it's sanitizing, okay? All of these mean there's some level of microorganisms that are left. Only the word sterile means we're getting rid of everything. All of these other terms simply mean we're reducing the number of microbes. So decontamination reduces the pathogens to a level considered safe. Degerming is simply using to decrease the number of microbes in an area, and degerming is typically used specifically for the skin. Sanitize, a substantially reduced microbial population. Do you see how there is very little difference between all three of these terms? They basically mean the same exact thing, okay? But it makes people feel better. We have decontaminated that for you. Okay? That could mean you sprayed some Lysol on it. That doesn't mean it's clean. That doesn't mean we've gotten rid of all of the germs that are on it. It simply means we've wiped it down. That's it. Okay? Same thing with degerming. Okay? Degerming sounds a little more nasty, so most people you're not going to see that term as much. But sanitize implies a substantially reduced microbial population. Okay? Yes, like your hand sanitizer. But you know what? You see this word, it's been sanitized, and people feel safe. Okay? Have you ever been to Chuy's at Easton? It's a Mexican restaurant over by Whole Foods. Their chips are amazing. They have good margaritas too. Okay? But they bring you the silverware. The silverware is wrapped in a little paper bag, basically. And it basically just means some random employee at Chuy's didn't put their hands all over it when they gave it to you. It says, this silverware has been sanitized for your protection. Okay, it means you washed it. You use soap and water, and then you put it in these little bags, okay? It doesn't mean they sterilized it. It doesn't mean they stuck it in an autoclave. It basically means we're using this to try and keep it nice and fresh. And it's totally enclosed, so nobody's hands are all on it but your own. But it doesn't actually mean in the grand scheme of things that your silverware is any cleaner there than it is anywhere else. But people see it and they go, oh, this is great. My silverware has been sanitized. I'm doing good. It's all in the mind, right? So all these mind games we want to play, okay? When you see this term preservation, preservation is almost the opposite. Preservation is basically delaying the spoilage of some sort of perishable item, okay? Typically, this means adding in any sort of growth inhibitor until we get to a right amount where this item is not going to go bad as quickly. That could mean adding in extra salt. That could mean adding in nitrates and nitrites. That could mean adding in other chemical preservatives, okay? You want to see what that's like? Go and look at a loaf of Wonder Bread at the store, okay? All of those loaves of bread all down those shelves. Pick one up and look at it. There are typically a whole bunch of chemicals in there you don't recognize. All of those are chemical preservatives. It says, look, we know this bread might be on the shelf for a few days before somebody buys it. And then we know it might sit in your house for a week. So we've added these chemicals to make sure our bread stays nice and fresh and soft until you actually go and eat it. Yeah?
How does that affect our bodies, though?
There's the question. Okay? So yes, we are focusing in terms of what it's doing to the microbes. But then what does it do to you is a whole nother ballgame. So nitrates and nitrites have been linked to things like cancer. They're in hot dogs. They're in all kinds of preserved meats. Bacon. Everybody loves bacon. Most people love bacon. Okay? It's all in bacon and other stuff. There are some natural nitrates and nitrites, but when you load in all of these chemical preservatives, yes, there is no guarantee that it's not affecting your body along with it. Yes. Yeah, so we can get into a whole debate on, what's better, all right? That nice fresh loaf of bread at the store, especially as it gets warm, might only last on your counter a couple of days. Can you eat a whole loaf of bread in a couple of days, or would you rather have one of these other ones that's going to stay and you don't have to worry about it, but it's got these extra chemicals in there? It's kind of a trade-off. Absolutely. So microbial methods are going to vary highly depending on where you're at. Guys, what you do at home is different than what we're going to do here in a lab, or what a research lab would do, or what would happen in a hospital or other care facility, okay, or water treatment plant. You're at home, your biggest issue at home is, am I going to get salmonella food poisoning from some, poultry product I ate, okay? Your levels of control that are needed are lower. You want to practice the 5-second rule at home, go for it. You're rarely going to get sick off of something like that at home. But you drop something on the floor, here in the lab, somewhere like that, you're not eating it. Toss that sucker in the trash, okay? So what you do at home versus your level of control somewhere else is going to vary. You go into a hospital and you're having surgery, you want to know that whatever it is they're cutting into you with is totally sterile, okay? But does your silverware at home need to be sterile? No. Yes, you're eating off of it, absolutely. But, hopefully, you're not stabbing yourself with it. It's not going in your body. Some small amount of bacteria that come in off of a fork you're eating off of at home, they're all going to die the minute they hit all that acid in your stomach, okay? So your levels of control are going to vary. What you use is going to vary. What we use here is going to be different than what you might use at home, okay? Trying to find the most effective procedure is complicated, and there's no one right or wrong answer. Every grocery store has that aisle, and it's a whole aisle, and it's all dedicated to cleaning products. There are 30 different kinds of like Tide, clothes detergent, okay? There are 10 different kinds of things for your dishwasher, all right? You can buy Dawn, you can buy all kinds of different soap. Mildew, you want to get rid of mildew? Look, there's like 10 different brands that do it. And guess what? Most of them all have the same active ingredients, okay? So there's no ideal method. And what works for you might be different than what someone else does, and that's fine. There's a lot of things that you have to consider when trying to pick what do I want to use for microbial control. You have to think about what types of microbes might be present. The types of microbes you have present in your house are going to be different from the types of microbes present in a hospital, right? At home, what do you have? You have a lot of soil and dust organisms. You have things that come in off of food that you have purchased and brought in, whether that's raw meat, whether that's vegetables. You're bringing in bacteria that way. If you wear your shoes around your house, walk all around your house with your shoes on, shoes you've worn outside, you're bringing in different levels of organisms, okay? The type of microbe is going to be different. If you are in a hospital setting, you can have some serious microorganisms given that everybody that's there is typically sick, okay? Or you're on a burn floor and you're concerned about Pseudomonas coming in on ornamental flowers, okay? Or you're trying to clean a ventilator, something that's had access to someone's lungs and they had pneumonia or something like that, okay? So the type of microbe is going to vary depending on your needs and your usage. Your extensive contamination is going to be different, okay? Have you ever sat and thought, when was the last time I washed that, okay? That's going to make a difference in terms of extensive contamination. How dirty is your sink? Did you clean it yesterday? Or has it been like 2 weeks since you scrubbed it with something, okay? How many organisms are there are going to differ. What is the environment? Is this something you can spray a chemical on, or is it some electronic that you don't want to get wet, you don't want to do something with it? Or can you put it under heat, or is it going to melt under a heat source? So the environment makes a difference. Also, the environment makes a difference thinking about what you're dumping down the drain, okay? Your risk of infection. In a hospital setting, the risk of infection is going to be higher, your risk of infection at home is going to be much lower. So there's different levels of control there. The composition of the infected item makes a difference as well. Are we just going to throw that away? There are times when I'm sure, you found something and you're like, you know what, that is just too nasty. We're just dumping the whole thing in the trash as opposed to trying to save it. Okay? It's all about picking the right control method and the right procedure. All right? You know, washing your clothes. You go to the gym, you got stinky, smelly clothes. Guess what? There's bacteria all over those. Okay? It's fine. You throw them in the washing machine. You use some soap. You put them in the dryer. A small amount of heat is going to kill whatever is on them. Okay? My washing machine has a sanitizing cycle. It superheats the water, and it runs for 3 hours. I've never used it in like 9 years. You know why? Because if my clothes are that bad that they need to go through a sanitizing cycle, I should just toss it in the trash. Okay, like why? And what is that going to do to your clothes by the time you superheat the water and you let them sit in there for 3 hours? Like, that just seems ridiculous. Okay? What were we going to say, Sophia?
We use tea tree oil
Yeah.
in our washing machine because at my house we all train jiu-jitsu and martial arts. So it's pretty nasty whatever you can pick on those mats.
Right. Exactly. Yeah. So when we get to antibiotics and stuff like that, we can talk more about natural products and essential oils and all kinds of fun stuff like that. But you're right, it's all about what do I have access to, what happens, what's going on, and what do I want to treat? How do I want to treat it? What am I thinking about?
I have a question.
Yeah.
So when you hand wash the clothes, does it mean you still have the bacteria in it?
So as long as you're using some soap and you're creating those micelles, like what we talked about with washing your hands, you're going to get rid of a large number of the microorganisms that are in there. And as long as you're getting rid of most food sources that would be on there as well. So if you're sweating in those clothes and everything else, there's enough of a nutrient source in there to keep those bacteria alive. If you're washing them and you're using some soap, you're removing not only the organisms, but you're also removing the food source as well. So things typically are going to die at that point. Yeah. The type of microbe you've got is important. We just went through that diversity section. Okay? Understand what you're gonna come in contact with at home versus what you're gonna come in contact with somewhere else. Most of the things you're trying to kill off at home are basic bacteria, basic vegetative cells. They're not resistant in any way, and they're gonna die fairly easily. Okay? Do you have things at home that might have some endospores on it? Yes, it's possible. Bacillus species are common ground organism species. They're common in soil and dust. It's possible that they're there. Most of the ones you're coming in contact with aren't going to make you sick, though, so it's not going to be that big of a deal. But whether you're talking about a cyst or an oocyte, okay, fruiting bodies, there's going to be a different level of resistance there. Mycobacterium, for example. We talked about how mycobacterium are acid-fast. You've got a patient in a hospital who's got a TB infection, who's been on a ventilator. That ventilator likely has Mycobacterium tuberculosis on it. You want to clean it, and you want to do a darn good job of cleaning it. And when you do it, you also have to understand that what you're trying to eliminate has a cell wall structure with all of these waxes and lipids that are going to protect it against most types of chemicals. So you better choose the right chemical to clean that ventilator in that situation. Right? So there are times in which you really have to consider this, and other times where you go, okay, I'm at home, most of what I'm coming in contact with is not going to be that big of a deal. Pseudomonas species. Again, common in burn victims, okay? Can cause a number of other infections. These guys are actually capable of growing on things like soap. They survive in the soap. So if what you think you have is Pseudomonas or it's present in that area, your level of control has got to take on or take into account that we've got this organism present that's going to require a little bit more stringent work in terms of getting rid of it. So you have to understand there's a lot of factors that go into what am I trying to kill and how do I try and do it. This is a nice table from your book because it outlines all kinds of various types of physical methods of control and how we would achieve them. So whether that's Heat. There's two different types of heat, using moist heat versus using dry heat. Believe it or not, moist heat is actually a lot more effective than a dry heat method. So throwing something in the oven is a dry heat method. Boiling something on the stove is a moist heat method. Okay? In class, in another week, we're going to test what the effects of boiling are on an organism. Okay? We're going to boil these tubes. We're going to take out aliquots of the organism, the culture, at various time points, and you're going to see how boiling affects those vegetative cells. And we're going to have a basic vegetative cell. We're going to have a cell that forms endospores, but it's a fresh culture. And we're going to have Bacillus subtilis, our old friend that forms endospores, but we're going to have a culture that has a lot of endospores. And you're going to see how that moist heat method affects those organisms. Dry heat methods. Anytime you take that loop in class and you stick it in the flame, you're incinerating whatever is on it. That's a dry heat method. Okay? You can filter things. Okay? How many of you have a water filter at home? Okay? You know, you're filtering those liquids to try and remove bacteria, contaminants, all kinds of things. Your furnace all has a filter on it. The finer the pore size on your furnace filter, the more fine particles you are able to filter out, including things like fungal spores and that sort of thing that might be in the air. You can use radiation. So we'll talk about UV light. We will talk about, bless you, we'll talk about ionizing radiation as well and how these are used for chemical control methods. You will do this in the lab. In the lab, we will expose bacteria to UV lights for various amounts of time, and then we're going to come back and look and see, okay, what is actually happening to this? How is it working? How's it not working? Can the organisms overcome that level of repair or not overcome that level of repair? You can buy little UV lights that go in a bottle of water. If you're traveling abroad, they sell them. They look like a little pen light. You stick it in the bottle of water. You turn it on. You move it around for a set amount of time. And it's to kill whatever is in the water. You can buy them where they go on your fish tank. So we can use all different kinds of methods to control microorganisms or to get rid of the growth of microorganisms. It's all about what you're doing and how is that achieved. When we say boiling something is heating it to 100 degrees Celsius. For example, you're asked to boil your water. If you don't actually bring it to 100 degrees Celsius, you don't reach that temperature. Think about what temperature is doing to those enzymes. The temperature has to be raised over the maximum in order to denature the enzymes. If you don't reach a good rolling boil, those mesophiles are not dying. Okay, so boiling is 100 degrees Celsius. It's going to destroy most microorganisms, most viruses. It's typically not an effective means of sterilization in that it does not destroy the endospores. So that exercise I talked about in class, the whole point of doing the exercise is to show you that we can kill something like staph or E. coli by boiling. But when we've got this Bacillus subtilis and it's formed a lot of endospores, the boiling doesn't kill it off. So even though we boil it for 10 minutes or 15 minutes, we still see the endospores that are coming up in the end. We still see growth of the organism in the end because we didn't inactivate the endospores. When you pasteurize something, Louis Pasteur is our good old friend. He developed pasteurization. He did this. He was very selfish. He did this because he was French and he liked wine. And his wine would eventually go bad. He was like, I got to find a method to keep my wine from going bad. And pasteurization ended up being it. Okay? It doesn't sterilize, but it significantly reduces the number of organisms. Okay? You buy that gallon of milk at the store. The gallon of milk is in the refrigerated section. It's kept cold because keeping it cold keeps those bacteria that might remain in a static state. Okay? The milk has been pasteurized. It's not sterile. The milk all has an expiration date on it. The expiration date is to tell you that this is the time in which we think we can no longer guarantee that any bacteria that might remain in this milk will not make this milk go bad. Okay? You took an unopened gallon of milk and you just let it sit. It is going to go bad. If you let it sit at room temp, it's going to go bad fairly quickly. If you keep it in the fridge, you've got a higher amount of time, depending on the level of pasteurization. Okay? You can increase or decrease the ability of the rate in which your milk goes bad. Think about it. You take the milk out, you pour yourself a glass, you pour it over some cereal, you put the milk back in. The longer the milk sits at room temp, the more that temperature goes up, and the more those bacterial enzymes function, which then causes the bacteria to multiply, which then increases the rate in which it goes bad. If you are someone who is drinking out of the carton on a regular basis, I don't care what you say, there are always going to be some bacteria from around. You can say you don't backwash or anything like that. You were good, okay? There are always going to be bacteria from your mouth that end up in that milk if you're drinking out of the carton. That is going to increase the rate in which it goes bad. But if you're someone who's drinking a lot of milk, sure, you might finish it before it actually goes bad, okay? But all of these things are to help you understand it's just pasteurized. We've reduced the number of bacteria, but it's not sterile. You buy the little aseptic packs of milk that are shelf stable. That is sterile milk. Okay, that's going to last a heck of a lot longer, which is why it doesn't need to be refrigerated. Okay, it is likely going to taste different because it's experienced a higher level of heat than the regular milk from the refrigerated section out of the carton. Most protocols for pasteurization lately. Most protocols are the HT-ST method. They are high temp, short time. You heat the milk to 72 degrees Celsius. You'll hold it there for 15 seconds and then you cool it down. You can also get ultra high temperature, in which case we've heated it to 140 to 150. We hold it there for several seconds and then rapidly cooled. The pasteurization method that's used is directly related to the amount of shelf life time on your milk. If you ever picked up a gallon and went, okay, this gallon expires in a week, and then looked at some gallons at the store and you're like, why is it that this expiration date is like a month down the road, okay? It has to deal with which pasteurization method they used. How big the batch size was when they pasteurized that milk, that makes a difference, okay? So your shelf life is directly determined by how long you're pasteurizing something, as well as what it is. Pressurized steam is a really good heat control method. So boiling something really good, steam under pressure is going to be capable of sterilizing most items. So in the lab, all of the media that you use, all of those things were all autoclaved before we poured everything. Okay, all of the media gets pressurized steam, and then we pour it into those individual plates before it solidifies. Okay, so when we use things in the lab, they've all been sterilized beforehand. Your dentist offices typically have a tiny little autoclave in there where they stick all of the utensils. It's steam. You heat this water, you make steam, it helps increase the pressure. It's a preferred method of sterilization. In particular, it's 121 degrees Celsius. So it's not that high, it's not that hot, but it's under 15 pounds per square inch of pressure, and you treat it that way for 15 minutes. So having that heat under pressure is going to be enough to inactivate the endospores. It's going to be enough to kill most things that are in there. Interestingly enough, if you had prions have to go into the autoclave at a higher temperature for 4.5 hours in order to be inactivated. All right, so remember back when we talked about those and how they cause spongiform encephalopathy. We don't really have normal control methods that are inactivating prions. Right? So we kind of set them aside. You should not be coming in contact with them anyway. We discussed you are not practicing human cannibalism, at least we don't think so. So those shouldn't really be an issue. But the steam under pressure is a really good way to sterilize versus simply heating something for a set amount of time as being a pasteurization method, because we're lowering the number of microorganisms. Dry heat is totally not as effective as moist heat. Okay? You can use your oven for sterilization. However, you're going to require a heck of a lot longer of time in order to do that. Okay? Typically what happens is we use, you can use something, it's called autoclave tape. You can also have it for a dry oven as well. The way you know something has been sterilized, you wrap it, you use this tape. When it's had enough time to totally kill everything and the pressure and temperature and everything else hit the right spot, this actually gets these little black lines on it. If it comes out of the machine and it doesn't have those little lines, it tells you it wasn't heated appropriately enough and we haven't actually sterilized anything that was on it, okay? So you can use dry heat, it just depends on what you're doing. Incineration is a dry heat method. It is reliable and it does work a lot faster, but you're basically burning something. You're sending everything to ash. So in the lab, we can take your inoculating loop and we can stick it in the flame and we can incinerate it, okay? But you try and do this at home, incinerating most items you have is probably not going to be good. You're going to destroy whatever it is, right? So you have to think about what is the level of control method that we can use. How do we want to do that? What's appropriate? Filtration is your other physical control method. Basically using a filter, the filter has tiny little pores. So you can see little pores that are all on this membrane here. Anything that's small enough to go through the membrane comes on out. Anything that's too big to go through the pores stays on up. As you vary your pore size, you can vary what's going through the membrane and what's not going through the membrane. So you can filter liquids, you can filter air. It really just depends. If you've ever tried to buy a furnace filter, okay, you can buy some that are $1 a piece. Super cheap. They don't really provide any sort of filtration method as well, at all. And then you can find other ones that are going to cost you $40, $50 that are going to claim to get rid of all the pollen and the pet dander, and for someone who's got allergies, this is going to get rid of everything that's in the air, okay? It's all about the pore size and what's letting things through. Likewise, if you never change your filter, that doesn't help you out, okay? These filters are going to get clunked up, basically, gunked up over time, and then they don't work as effectively, and you're going to get pass-through of organisms since everything gets pushed against it. What are we thinking so far? We making sense with a lot of this stuff? Okay. Radiation is another method. Radiation is not a method that you're going to use frequently at home, but it does work well for microbial control nonetheless. And you've got two different types. You have ionizing versus non-ionizing. Here is where your understanding of chemistry is going to come back in this situation. So ionizing radiation, remember, in an atom, atoms have protons and neutrons in the nucleus, and they have electrons which are around the outside orbiting around. What happens in ionizing radiation is that this type of radiation is actually going to eject orbital electrons from that atom. And it causes an ion to form. So an ion is an atom that has simply lost its normal number of electrons. Okay? So ionizing radiation is an interesting form of radiation that's used for microbial control, because ultimately it causes breakages in the DNA of any living cell. Think back to your replication. When a cell needs to divide, whether it's going through binary fission or mitosis, one of the first things it has to do is to copy its DNA. So it goes through DNA replication, and then it replicates all of the parts it needs in order to divide. What happens in ionizing radiation is that you create all of these breakages in the DNA. Guess what? Replication doesn't happen when your DNA is chopped into all kinds of little bits. It also makes transcription and translation a pretty big problem as well. Okay? So cutting apart a cell's DNA is a good way to lead to microbial death because the cell can't replicate anymore. Remember what we said a few slides back. A cell is in a state of death if it can no longer reproduce under ideal conditions. You chop apart their DNA, that is not ideal conditions. They are not going to be able to replicate anymore. Okay? Non-ionizing radiation, on the other hand, simply excites the atoms. And what this does is it actually creates bonds between certain bases on the same DNA strand. So this creates abnormal bonds. Abnormal bonds, too many of those as well, alter DNA replication, which alter growth of the organism. So whether you're using ionizing or non-ionizing radiation, you're still going to cause problems with the DNA, enough to affect microbial growth. Interesting to note here, ionizing radiation is okay, it goes through all kinds of barriers, but non-ionizing radiation is not capable of penetrating a barrier. Non-ionizing radiation is the same thing as saying UV light. So UV light is not good at going through barriers. Think about your sunscreen. Apply sunscreen, you reapply sunscreen, you get the sunscreen and it's got all of these different SPF values. The SPF values relate to how much of the UV light it's blocking. That simple barrier layer that is the sunscreen that goes on your skin is enough of a barrier to make sure a certain amount of the UV light doesn't penetrate. Yes, you have to keep applying to make sure you keep up with that barrier, okay? You got clothes on? Your clothes are a good barrier at preventing UV light from going through, okay? You know UV light affects your cells just like it affects microbial cells, okay? Leads to uncontrolled growth, leads to all kinds of problems, damage, not good, okay? When we treat it against microbial cells, we can use it to kill them if we cause enough damage. Here's another way of looking at UV light. So UV light creates what are known as pyrimidine dimers, and your pyrimidine bases of your DNA are those C and T bases. Okay, this is again bringing back your basic biology. So anytime you get two T bases next to each other, or two C bases next to each other, or a C and a T base next to each other, they're going to be able to form dimers when you have the UV light present. Okay? Notice that it does it this way. It's doing it on adjacent strands. So yes, the bonds would normally be across, connecting opposite strands of DNA. When you hit them with UV light, the UV light creates these dimers, which then link bases on the same strand together. This as well creates a problem for transcription and translation because when those components come in, those polymerases come in, and they have to sit down and read the bases, 3 bases at a time, this messes up with that process. So it ultimately does lead to issues with microbial growth as well. What do we think about physical methods of control? We're doing okay? These are things you've used in most cases. Again, UV lights are common. You can get a UV light that goes on your fish tank, and as the light passes through from the filter, it passes through this UV light to kill algae and bacteria that are in the water. Okay? You can get them to help kill off bacteria in a water bottle. If you get a fancy electronic toothbrush, some of them now actually have these little UV housings. You put the top of the toothbrush in there, you close it up, and the UV light's supposed to kill all of the bacteria on your toothbrush, okay? I would probably tell you that it's not as reliable as you might think it is because of all those bristles and how it doesn't go through barriers and all that kind of stuff. But if it makes you feel better, yes, it is going to get rid of some level of bacteria on there, okay? Now we're going to get into chemicals as control. Right? So this is adding, anytime you're adding any sort of chemical, now we're moving into a chemical control method. Chemical control methods are important, and it's important as well that we understand what they're affecting. So in particular, we can affect proteins, DNA, or the cell membrane. Remember the terms. Remember it's in a state of microbial death when it can no longer reproduce under ideal conditions. And then think about back to our binary fission, and think about our metabolism. Think about the structures we said, what they do, how they're important. Okay? So DNA. Is DNA a vital cellular component? Yes, right? DNA goes to RNA, which goes to protein. Lots of things inside the cell are made of proteins. If you have no good intact DNA, you're ultimately going to affect your RNA and you're ultimately going to affect a cell's proteins. In addition, if a cell is going to reproduce, it's going to go through binary fission. It needs to copy its DNA first. If its DNA is chopped into little bits, guess what? That copying process doesn't occur. So if you alter a cell's DNA, enough, you cause enough damage, you're going to ultimately affect microbial growth. What are enzymes made of? Protein. You mess with microbial proteins, you're messing with microbial enzymes. Remember, if enzymes aren't working at their best or at their required rate, a cell doesn't complete chemical reactions fast enough to support its growth. Again, if it doesn't grow, it doesn't divide, it's in a state of microbial death. Proteins are also embedded in the membrane, in the cell wall. We talked about this, that fluid mosaic model. Remember how we said proteins act as gates and receptors, so they allow nutrients in and waste out. You're affecting all of these proteins, nutrients aren't coming in. Waste isn't going out. So messing with a cell's proteins affects it in all kinds of different ways, which ultimately leads to issues with microbial growth, which ultimately sends it into a death state. And then think about that cell membrane. Remember that cell membrane is that innermost barrier. It's what really regulates movement in and out of the cell. What happens if a cell's cell membrane is destroyed? What happens to that cell? It lyses. It's dead. It's a goner. Okay? So chemicals that are reacting with vital cellular sites ultimately lead to cell death because it's everything to control the growth of an organism, the division of a cell, and how that works. We're not going to go through all of the chemicals that are listed on here, but there are a number of them that I want to talk about in more details. Okay? And I'm leaving this on, I've left this on the slide to help you understand what they're affecting. So when you use alcohols or halogens, halogens is bleach, think bleach, okay? When you use some of these chemicals, you're affecting proteins. Remember, proteins are in the membrane, proteins are in the cell wall, proteins are used in everything inside the cell, including for enzymes. In lab, we spray that quaternary ammonia compound as a disinfectant when you walk in, before you leave. It affects the cytoplasmic membrane of these cells, and that's how it works as a disinfectant. Okay? So there are lots of different things that you can use, chemically speaking, but ultimately you're altering microbial control because they have very specific targets on the cell. It's not just this alcohol destroys the cell, it's how does it destroy the cell. That alcohol is affecting proteins. So what makes up proteins, how are proteins important, it's taking it a step further. Okay? These are all typically germicidal chemicals. Remember, germicidal basically is no real level of control. It simply means we're killing some level of bacteria. Depending on the chemical, that could be simply a disinfection method, or it can be a sterilization method, depending on what you're using and how you use it. If something is a sterilizing compound, but you don't leave it on long enough, it's not going to sterilize. Okay, so just remember there's a lot of factors that go into this. How potent these chemicals are is going to differ. All right, and guess what? You're never going to be able to pick something up and know from the store exactly how potent it is. Okay? You know bleach does a good job, but you don't know that it's actually killing absolutely everything you touch. Okay? Yeah, your can of Lysol could say kills 99.7%. And if you look in the fine print, it says of household germs. What on earth does that mean? What does that? That means nothing in the grand scheme. Potential household germs. It means they tested it on ones that they claim could be around your house that it could be killing. It doesn't mean it kills everything. It doesn't mean if you get some random thing around your house, the Lysol magically gets rid of it all. It doesn't mean that at all. Okay? Most of your chemicals are all regulated. Your antiseptics are all regulated by the FDA. Look on anything you have at home. Primary example of this, pick up a bottle of mouthwash. You have a bottle of mouthwash, it will have a little stamp on it showing you it's been regulated by the FDA. All of the disinfectants are regulated by the EPA, the Environmental Protection Agency, because we want to see when those go down the drain, what kind of effects are we having on our environment, okay? Yes, we kill bacteria, but what else are we doing along with it, okay? Your germicidal agents, or these chemicals, are typically grouped according to their potency. So a sterilant is going to be capable of killing all microorganisms, getting rid of endospores. It makes things sterile. Again, if it doesn't say sterile, that means there's some level of microorganisms left behind. You technically have 3 different levels of disinfectants. High-level disinfectants destroy most things, but not endospores. Intermediate level disinfectants destroy lots of different types of vegetative cells and fungi, not all viruses, and again, not the endospores. And the low level of disinfectants are going to get rid of most of the easy things to kill, but not anything with any sort of level of resistance. Things like mycobacterium, which we talked about last time. Naked viruses, remember viruses that only have the capsid that are missing the envelope are actually harder to kill than enveloped viruses. So this helps us understand where our diversity comes in. It also goes back to our understanding of viruses and how those viruses work. Again, you're not going to pick something up and go, is this a high-level disinfectant, an intermediate-level disinfectant, or a low? It's not going to stay on there. Understand that the disinfectant is not killing everything, right? How do you choose the right chemical? How toxic is it? You know, guess what? Bleach works really good, but bleach is super toxic, okay? So you want to dilute it down. You want to think about what you're dumping it in. The activity in the presence of organic matter. Most chemicals are going to be less effective the more organic matter that's present, okay? So you want to clean your sink. Your sink is full of a bunch of food particles in the bottom of it. Guess what? All of those food particles are going to affect the activity of whatever it is you're dumping in. Try and clean it first, mechanical removal first, then add in the chemical, and it's going to be much more efficient. What's working with a residue? Okay? Bleach leaves a residue, which is why they tell you if you're using it to kill anything, you want to rinse it again afterwards. Your baby toys or stuff like that, things that are outside, you can use a bleach solution. However, you want to rinse it afterwards because it does leave behind a residue. Cost and availability. This disinfectant could be the greatest thing in the world, but if it's $100, nobody's buying it. If it's too expensive, you're not going to buy it. Think about, I used to go to the store. I used to get these coupons for Lysol products. And I'd get them, and they'd be double coupon, and I'd wait until they had it half price. I'd get them for literally like pennies. And I just, in that respect, I became one of those like crazy coupon ladies where they have like a million. It was I didn't care what brand it was. All I cared about was that I got it and it was super cheap, and it all has the same chemicals in it now. So what's the big deal? Okay, I no longer do that. I use more natural products on everything. But cost and availability makes a difference. Storage and stability. If you've got an item and it's no longer good after a while, that makes a difference. Guess what? That gallon of bleach loses its potency the longer it sits. Okay. Environmental risk makes a difference as well. What are you dumping down the drain and what is it affecting? Okay? So there's a lot of things that go into how to choose the right chemical and thinking about what it's going to do and how it works. Again, here's another table. I like the table because it breaks apart the chemicals and helps you understand examples and then what it's affecting. So whether that's alcohols, aldehydes, the biggest one that's in here that you would have heard of is formaldehyde. Okay? Understanding what you're going to have access to and what you're not going to have access to. A lot of these more stringent aldehydes are used in a medical setting only. They're not available for home use, depending on what you've got. Halogens are common. So most of you, I would bet, have bleach somewhere, okay, that you can use for microbial control. So understanding what you're doing and what it's affecting is important. You can use metals as well. Have you ever seen Band-Aids that have little bits of silver strung through them? The silver is actually an antimicrobial compound all on its own. Okay? My favorite cleaning compound happens to be hydrogen peroxide. Okay? It's a peroxygen. It works by basically creating these toxic oxygen radicals that kill most things. Right? Triclosan. Some of you may have heard of this. This was in all kinds of antibacterial hand soap. It's now banned by the FDA. All right? But it's a phenol-based compound. Okay? So lots and lots of different chemicals have all kinds of different effectiveness. Alcohols in general, you want a solution of anywhere from 60 to 80% isopropanol or ethanol. So those are the two alcohols that are going to be effective. You're not using your tequila at home to try and kill bacteria, okay? Ethyl alcohol, isopropanol, it needs to be between 60 and 80%. How are these going to be effective? They're ultimately dealing with proteins. They're going to affect proteins specifically and enzymes. They typically damage lipid membranes as well, because remember, the lipid membranes are all going to have embedded proteins and other stuff in there, okay? Alcohol can be used as an antiseptic or a disinfectant, but understand its limitations. It evaporates quickly. This is why when we talked about the hand sanitizer, said, hey, you've got these hand sanitizers, they have alcohol in them. Your hands need to stay wet for 30 seconds. If they're drying faster than that, you're not leaving them on long enough to do any good because it's evaporating too fast, okay? Alcohols can be harsh, they can be drying, so you do want to understand what you're putting it on. Bleach. Bleach is great. It oxidizes proteins. Oh, here's a good connection. What do we remember when it says oxidize? What is that doing to the electrons? So, right? So I always end up thinking about it backwards. If we are reducing something, it's gaining electrons. So if we're oxidizing something, it's losing electrons. So in this case, its effect in oxidizing proteins is to take those electrons and move them around, to lose electrons from the protein. Examples of halogens, and we don't have a periodic table on here, but the halogens are a certain category down the periodic table. It happens to include two common disinfectants, which are chlorine and iodine. That chlorine is going to kill all types of organisms and viruses. It's a really good disinfectant. However, it is completely caustic to your skin and mucous membranes. You ever had bleach on your fingers and they feel slimy? Yeah, that's actually liquefying your cells. That's not good. You want to rinse that off as quickly as you can, okay? It's basically doing that to the bacteria as well. So it works really good, but fairly toxic in the grand scheme of things, okay? Iodine. Sometimes they use iodine, you'll see it before surgery or before they put in a needle, something like that. Good at killing vegetative cells, not real reliable with endospores, okay? Typically it's mixed in a tincture with alcohol normally, but it can be used with other components to dilute it down as well for use on skin. So disinfectant as well as antiseptic, depending on how you've used it. Iodine turns everything brown, okay? So while iodine works as a good disinfectant, chances are you're not using it at home because you don't want it to turn everything brown, okay? Metal compounds work, believe it or not, really well. Some organisms are going to do a better job than others, depending on what it is, and it has to be the right compound, okay? High concentrations of metals are typically toxic to human tissue, but in small doses, these are typically okay. Silver is still really commonly used as a disinfectant. Again, if you look down the bandage aisle, you can typically find silver in creams. You can find silver in Band-Aids as well. It's a common disinfectant. What you're looking at here is we've got a silver amalgam on the left and a gold foil on the right. All of these tiny little white dots on this plate are all little colonies. So what you see here is we've got a general zone of inhibition where we're inhibiting lots of organisms. All of the tiny little dots that are popping up in this general circle are all resistant to that gold foil. This silver amalgam has less resistance to it. It has a bigger zone of inhibition as well. This one is working better against whatever this organism is. Silver amalgams used to be used for fillings when you had cavities. They'd put a silver amalgam, stick that, shove that down in the tooth. Yes, it was really durable and it was solid. It also had antimicrobial properties that were helpful. Yes, now they've come out and been like, oh, it's toxic, it's leaching stuff in, you want to get all that out of your body. There's other things that happen to that, but from a microbial perspective, it does do a good job at killing off microorganisms. Peroxygens are on here. Yes, I said peroxide is like my favorite cleaner of all time. I use peroxide everywhere. I go through lots of it, okay? It doesn't leave a residue. What happens here is that hydrogen peroxide breaks down into water and oxygen gas. It basically creates toxic oxygen radicals by making that oxygen gas on there. And only organisms that are capable of producing enzymes to break that down are going to be able to survive. In large doses and specifically if you heat it up as well, this can actually be a sterilizer. Okay, so typically when you buy hydrogen peroxide from the store, hydrogen peroxide, that bottle that you buy is 3% hydrogen peroxide. Higher concentrations of hydrogen peroxide are good and work really nicely to actually sterilize things. You can't buy it. We don't have access to buying anything like that. But it does work really well. It doesn't leave a residue, and it's going to kill most things. You want a fun science experiment, pour some hydrogen peroxide in your kitchen sink, okay? It bubbles. It bubbles because there are bacteria in the sink, okay? What breaks it down is the catalase enzyme. So if you ever use peroxide and you've dumped it on a wound and it bubbles like that, it doesn't necessarily mean that you've got bacteria there. Your living cells actually make the catalase enzyme that break that down. Okay, so that's why it does bubble when you put it on your skin as well. But you can use it as a mouthwash. You can use it in all kinds of different ways. Okay, it does a good job cleaning. Peracetic acid is another peroxygen. It's more potent than hydrogen peroxide. It's really effective on organic material, but typically this one's not used in home disinfecting. But another example. When you see these phenol-based compounds, phenol is an active ingredient in Lysol. Okay? Phenol, its other name is carbolic acid. Carbolic acid doesn't sound as nice as phenol. Carbolic acid kind of sounds kind of toxic. So typically they say it's a phenol-based product. This is actually one of the earliest known disinfectants ever used. However, it's totally toxic. You should not be breathing this in. Inhaling this into your lungs is really not good. Lysol is okay because Lysol has such a teeny tiny amount of phenol that it's basically considered not an issue. But what it does is it destroys the plasma membrane. It also denatures proteins, so affecting microbial growth because they basically lyse open or they cannot replicate. They don't do enough damage to the plasma membrane. Lysol is good in killing most vegetative cells. Mycobacterium in higher concentrations, but not reliable on all groups of viruses. What I want you to understand is it's a disinfectant, which means we're not killing everything. Don't spray that Lysol and feel like it's some magic potion that eliminates all kinds of bacteria, because it doesn't. Triclosan, hexachlorophene phenols, these are not as common anymore, but they were used in soaps and lotions. Because it's on the skin, you're thinking antiseptic. Used in toothpaste, lots of different brands of toothpaste had it as well. The FDA basically came out and said they had until the end of 2016 to prove that these chemicals were not harmful. And any company that couldn't prove that the triclosan wasn't harmful to humans had to remove it from their products. So Bath and Body Works had to take it out of all of their antibacterial soaps. All of the toothpaste companies, with the exception of Colgate. Colgate had done a study like 10 years earlier that they claimed that the FDA managed to say, yeah, okay, you're fine. They were allowed to leave it in. Everybody else removed it. Okay, I would argue that they should be taking it out anyway. But triclosan is a known antibacterial compound. It kills a lot of organisms. Keep in mind with any of these, I'm not saying they're safe for you. I'm not saying they're good. You know, don't drink bleach. That's not a good thing to do. But understand what we're focusing on, what they're doing to bacterial cells. In terms of, so we've talked about disinfectants. The opposite side of the coin is to talk about preservatives, or adding chemicals in to try and prevent organisms from going bad. Some preservation methods that don't require chemicals. Reducing all the available water, freezing them, low temperature storage. All of that is going to be a good preservative method, all right? Your beef jerky. You buy beef jerky at the store, we've removed all the water. So it's a preservation method because we've removed all the water. Now typically it's all treated with nitrates and nitrites and other stuff as well. But they're doing their part to try and remove the microorganisms. If you see things that have formaldehyde in it, phenols in it, it's basically a chemical preservative. You've seen there's a study on Diet Coke, diet sodas in general, that basically if they get warm, part of the breakdown product produces formaldehyde in there. So they're like, look, the acid is basically a preservative. Go in the anatomy lab, okay? Smell the formaldehyde. It's all a preservative, whether you're preserving the body or you're preserving something else, okay? Weak organic acids are good food preservatives as well. So ascorbic acid is, anybody know? It's a nice vitamin C, okay? Adding a little bit of acid is a really good food preservative. Propionic acids are used in a lot of cheeses, okay? So adding acids to things, a slight weak acid, is a really good preservative method as well. Prevents bacteria from growing. Again, think about that pH in our discussion there, okay? Nitrates and nitrites are used in all kinds of processed meats. All you gotta do is look on the package. The reason we do that is because it inhibits germination of endospores. It also inhibits growth of vegetative cells. So all of these processed meats can be sitting for a certain amount of time. Adding in all of those preservatives helps to increase the shelf life here. Important to note, those have been shown to be a fairly potent carcinogen. So you kind of have to balance the amount of nitrates and nitrites. Yeah?
How long would they last if they didn't have the preservatives in them, like naturally?
Not days tops. Yeah. I mean, maybe, depending on how open they are and what the temperature is, you could be talking 48 hours or less. You could get another day or two out of it, depending on what it is. Think about the difference between fresh meat versus getting something like lunch meat. The fresh meat's going to go bad fairly quickly. The lunch meat you could have in your fridge like a week and be okay. You know what it is. Yeah. How many more slides do we have? 3? Okay, let's just finish it. Getting rid of the water is a good preservative method. All right, you buy a box new shoes. They've got those little desiccator packets in there. Silica gel says do not eat all over it. It's meant to remove the water. Anytime you dry something out, you're going to preserve it for a certain amount of time, okay? Yes, salt is a way of decreasing water availability. Understand that some organisms can survive in high salt methods, so it really just depends on what you're using. Freeze-dried foods. Your coffee, believe it or not, has all been freeze-dried. So they freeze it as well as removing all the water, and that process really does eliminate all the bacteria that are in there. So it's a preservative method, but it's a method of not adding any chemicals to it. You're not memorizing the table. Don't do it. What I'm pointing this out for, this is from a different textbook, is to show you that a lot of these things all have the same compounds. So whether you like Tilex versus Lysol, or you like the scent of this one, or this one was cheaper, it doesn't matter because typically the root chemicals are all the same. Okay, so if you're ever wondering what's in something, yes, look at the label. Typically when it comes to something like these chemicals you're going to find in the store, looking them up online is going to be better than trying to find a list of chemicals, because most of the time the ingredients are not fully on the label. If you want to choose a chemical, what do you want to keep in mind? You want it to kill most organisms. You want it to act fast. You want it to penetrate inanimate surfaces. You want it to sanitize and deodorize. So all of those things that we talked about are variables, are things that you take into account when choosing a chemical. And there's no right or wrong reason. Think about it. Does this require sterilization, or is disinfection adequate? Your silverware doesn't need to be sterile. Tossing it in the dishwasher or washing it in the sink with some soap and water is enough to get rid of what's on there. Is it going to be reused, or is it going to be permanently discarded? If you're going to permanently get rid of it, what control method do you really need to use? Okay? Don't put plastic in the oven if you're going to, you want to do microbial control. The heat is probably going to melt it before you kill off whatever is on there. So you've got to really think about what you're choosing and what you're doing to it. Sound good? All right, let's take a 10-minute break.